PCR or LFP investigation of TLR4 gene mutation.
PCR stands for polymerase chain reaction. It is an important step for many labs regarding the investigation of genes and DNA. It is based on the nature of DNA polymerase and temperature. This reaction helps our DNA samples to be duplicated in a large amount, which can be used for further examinations. Read the instructions. Prepare DNA samples and solutions. Wear your lab coat. Tie your hair. Wear gloves before touching the samples. Lab safety is the priority. Adjust the pipette. We practiced how to use these equipments before getting pro at them. Label the samples. Relocate the DNA samples into new tubes.
Add different reagents to the PCR tube.
Mix the reactants. Adjust the PCR machine with correct temperatures and heating cycles. Oops. LOL, we named our file. Seal the PCR machine and we are ready to go. Our DNAs are duplicating.
What's happening inside?
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle.
Three hours later. Part 2. Think of DNA as a very, very thin thread, and the enzymes are the scissors that can cut the thread into pieces. Different people have different genes, so we'll get different number of fragments showing the genotype. Back to the experiment. Use restriction enzyme to cut our DNA. Set the optimal temperature for it to cut. Give it an hour. Prepare distilled water. Make the gel. Pour it into the mold. Remove the comb. Place the gel into the gel chamber. Let the running buffer fully cover the gel. Load different samples. Make sure change pipette tips between each sample to avoid cross-contamination. Run gel electrophoresis. This is what we got. The samples are AA homozygous.